Several methods of genetic modification have been covered in the previous videos. In this video, you'll find some points not covered before, along with questions to review the topic. To change an inherited trait using genetic engineering or gene editing, the genetic determinants of the trait and their sequences must be known. Only then can one decide how an individual can be engineered for benefit. Should a gene be knocked out? Should a gene be inserted? Or should a gene be edited? For example, the BCL11A gene was knocked out to treat sickle cell disease, enabling the fetal gamma chain of hemoglobin to be expressed instead of the beta chain of adult hemoglobin in individuals post-birth. The cry gene from the bacterium Bacillus thuringiensis was introduced into the corn genome to provide it with natural resistance against specific pests. The carbamyl phosphate synthetase 1 gene was edited to treat individuals with a deficiency of this enzyme resulting from a single nucleotide polymorphism. Scientists' hard work in research and their ingenuity underpin these technological developments. Wow! Scientists also contend with problems such as identifying the specific cells of the body to be targeted for trait alteration and how to deliver the change to those particular cells. For instance, to treat a blood disorder like sickle cell anemia, blood stem cells could be targeted in an ex-vivo setting. To address a vision problem, specific cells in the eyes may need to be targeted in vivo. Delivery methods include gene gun, injection, viral vectors, and lipid nanoparticles. Selecting a delivery method hinges on its appropriateness for the desired genetic modification, taking into account elements like insert size, immunogenicity, and expression duration. Once a genetic change has been attempted, it needs to be validated and the organism screened for off-target effects. Although validation by obtaining sample cells and sequencing the DNA or performing PCR assays is usually the best approach, in some cases this method may cause harm. Therefore, the benefits must be weighed against the harm. Observing the organism for the desired therapeutic effect and the absence of any adverse side effects or new disease symptoms can serve as an initial, albeit less precise indicator that the gene edit was successful and safe. Let's proceed to review the content with some questions. Question 1. Which of the following scenarios is best addressed by the specified genetic engineering technique? A. Precisely inserting a specific DNA sequence of several hundred base pairs at a target site in a mammalian cell genome, base editing. B. Introducing a large gene for insulin production into bacteria, traditional genetic engineering using plasmid introduction. C. Making a small insertion or deletion up to a few dozen base pairs with high precision and without a double-strand break, traditional genetic engineering using plasmid introduction. D. Correcting a single-point mutation, say A to G, in a human gene without double-strand breaks, traditional CRISPR. The correct answer is B. Traditional genetic engineering using plasmids is ideal for introducing whole genes or large DNA sequences into an organism, such as for protein production in bacteria. A is incorrect, as base editing is used to correct single nucleotide polymorphisms. C is incorrect, as the description given in C is more appropriate for prime editing. D is incorrect, as the original CRISPR-Cas9 technology uses double-stranded breaks. Question 2. A researcher successfully introduces a desired gene into mouse embryonic stem cells via homologous recombination. These genetically modified embryonic stem cells are then injected into blastocysts, which are subsequently implanted into surrogate mothers. Chimeric mice are born exhibiting various degrees of chimerism. Which of the following is the most critical factor determining whether these chimeric mice will produce progeny that carry the introduced gene. A. The sex of the chimeric mouse. B. The percentage of embryonic stem cell-derived tissue in the somatic organs of the chimeric mouse. C. The presence of the introduced gene in the germline, sperm or oocytes, of the chimeric mouse. D. The health and longevity of the chimeric mouse. E. The specific genetic background of the recipient blastocyst. The correct answer is C. The presence of the introduced gene in the germline, sperm or oocytes, of the chimeric mouse is the most critical factor. For the gene to be transmitted to the next generation, the genetically modified ES cells must contribute to the formation of the germline cells of the chimeric mouse. If the ES cells do not colonize the germline, the progeny will not inherit the modified gene, regardless of the presence of chimerism in other tissues. A is incorrect. While the sex determines how the gene is passed on, it doesn't decide whether or not it's passed on at all. Both male and female chimeras can transmit the gene if their germline is chimeric. B is incorrect. While a higher degree of chimerism in somatic tissues often correlates with germline chimerism, it's not a direct guarantee. A mouse can have significant somatic chimerism but lack ES cell contribution to the germline. D is incorrect. While essential for breeding, a healthy and long-lived mouse will not transmit the gene if its germline is not derived from the modified ES cells. E is incorrect. While genetic background can influence ES cell integration and chimerism efficiency, it's not the most critical factor for germline transmission once chimerism is established. The presence of the gene in the germline itself is paramount. Question 3. A researcher wants to investigate the role of a specific gene in neuronal development, but needs to ensure that the gene is only inactivated in mature neurons and not during earlier developmental stages or in any other cell types. Which genetic engineering system would be most appropriate for this controlled gene manipulation? A. CRISPR-Cas9 gene editing. B. Random insertional mutagenesis. C. RNA interference. D. Cre-Lox recombination system. The answer is D. The Cre-Lox system is specifically designed for conditional gene manipulation, allowing for gene inactivation or activation in specific cell types and or at a specific time point during development, making it suited for the researcher's needs. Question 4. To produce gene knockout mice, gene targeting via homologous recombination was attempted. The gene targeting vector used has the neomycin resistance gene, which confers G418 resistance, placed within the homology arms, while the herpes simplex virus thymidine kinase gene, which confers sensitivity to ganticlovir, is positioned outside the homology arms. Considering that both homologous and non-homologous recombination took place, which of the following statements is not correct about the possible outcome of the experiment? A. Cells with non-homologous insertion will be sensitive to ganticlovir. B. Non-recombinant cells will be sensitive to G418 and resistant to ganticlovir. C. Homologous recombination will ensure that cells will be resistant to both ganticlovir and G418. D. Homologous recombinants will grow on G418 containing media and will be sensitive to ganticlovir. The answer is D, as the question asks for the incorrect statement. Homologous recombinants integrate the neo-R gene, making them resistant to G418, so they will grow on G418 containing media. However, they lose the HSV-TK gene, which means they will be resistant, not sensitive, to gain cyclovia. A is correct. Non-homologous recombination results in the integration of the entire exogenous DNA construct, including the HSV thymidine kinase gene. Since the HSV thymidine kinase gene confers sensitivity to gain cyclovia, these cells will be sensitive to gain cyclovia. B is correct. Non-recombinant cells have not taken up the neo-R gene, so they will be sensitive to G418. They also lack the HSV thymidine kinase gene, which makes them resistant to gain cyclovia. C is correct. In homologous recombination, the neo-R gene is integrated, making cells resistant to G418. But the HSV thymidine kinase gene, located outside the homology arms, is lost. Therefore, homologous recombinants will be resistant to gain cyclovia. So they will be resistant to both. Question 5. A researcher is aiming to deliver a large gene, approximately 8 kilobases, into both dividing and non-dividing human cells for long-term expression with a high safety profile due to potential clinical applications. Which of the following viral vectors would be the most suitable choice? A. Adenovirus, B. Retrovirus, C. Adeno-associated virus, D. Lentivirus. The correct answer is D. Lentivirus. Lentiviruses can efficiently transduce both dividing and non-dividing cells, integrating their genetic material into the host genome and leading to long-term expression. They also have a relatively large packaging capacity of up to about 10 kilobases, which can accommodate an 8 kilobase gene. Furthermore, they generally have a better safety profile compared to adenoviruses in terms of immunogenicity, making them a strong candidate for clinical applications. A is incorrect, as adenoviruses, while effective for delivering large genes and transducing both dividing and non-dividing cells, are known for triggering strong immune responses and are generally associated with short-term expression, making them less ideal for long-term clinical applications. B is incorrect, as conventional retroviruses can deliver genes for long-term expression but require cell division for integration into the host genome, which makes them unsuitable for modifying non-dividing cells. They also carry some risk of insertional mutagenesis. C is incorrect, as adeno-associated viruses, while an excellent choice for safety and long-term expression in both dividing and non-dividing cells, has a small packaging capacity, typically around 4.7 kilobases, which is insufficient for an 8 kilobase gene. If you found this video helpful, please give it a thumbs up and subscribe. Your support is appreciated.